So it's a great honor and pleasure to have today introducing Professor Dr. David Julius and Professor Dr. Ardem Pateputian, who were awarded 2020 Kavli Prize in Neuroscience, and later in 2021 they were also awarded Nobel Prize in Physiology and Medicine for their discoveries that help us to understand how temperature, pain, and mechanical stimuli are converted into electrical impulses in our nervous system. So welcome. Our first speaker is going to be David Julius. David was born in Brighton Beach in Brooklyn, New York. Later, he studied at MIT, and after his studies in MIT, he joined the lab of Jeremy Torner and a Nobel Prize winner, Randy Shackman, at UC Berkeley, where he worked in mechanisms of peptide hormone processing and secretion in yeast. David later did his postdoc in another Nobel Prize winner, Richard Axel's lab at Columbia University, where he actually cloned and identified and studied the genes that are encoding serotonin receptors. David is currently a professor at UCSF and a trustee of Howard Hughes Medical Institute. Please join me, welcome Professor David Julius.
You guys have a seat. All right, thank you so much. It's great to be here today. It is true that I worked for two Nobel laureates, but I worked for them many years before they got their Nobel Prize, before they were over the hill. So anyway, all right, so it's great to be here today. I have this title that's a little bit off because I decided this morning that I'm tired of giving a retrospective talk. You can see that in the Nobel lecture, and instead I talk about something that's sort of new that we're doing. It's a bit of a work in progress, so forgive me if it's a little rough, but I thought that might be more interesting for this group than just going back over old history. All right, so, but nonetheless, I'm going to put in a few background slides so that you can see the neck of the woods we're in. All right, so I think today what you'll hear in the first session is about somatosensation, both what Ardem and I have worked on. And somatosensation is, is the sense that we colloquially refer to as our sense of touch. But it includes a number of submodalities, including touch, which Ardem will talk about more in his lecture, I presume, detection of mechanical stimulation to the body, proprioception as well, which he's worked on, detection of muscle displacement of muscle and joints, and then thermosensation, something that we've done a lot of work on that I won't touch on too much today, our ability to detect cool and warmth, and then nociception, which is really, in some ways, all of the above in the extreme, that is encompassed within this submodality that we call that leads to pain, cognitively pain. And my labs focus primarily on the aspects of somatosensation that are involved not just temperature sensation, but also in the context of nociception and pain, and that's really an area that we're interested in. So understanding pain. Why do we study pain? And that's because the pain system acts as our main warning and protective system that tells us when we have or are about to experience bodily injury and initiate appropriate protective reflexes. And I've said in one of these videos you might have seen, and I do believe this, that among our senses, I would say that the somatosensation and pain is the one that's most critical for our survival and well-being. And you can see this in people in whom genetic mutations render these systems inoperable. People who cannot sense pain are great risk of injury, and children who do not have this modality don't learn how to protect themselves and are frequently injured or even die as a result of not understanding the world that they live in. The problem is that pain can outlive its usefulness as an acute warning system. We all know that, especially those of us over 50, and instead become persistent and debilitating. And one of the big challenges in this field is to really understand how this switch occurs, with the goal of either preventing or reversing it. So one question is, how do different chronic pain syndromes differ? We use, and I'll come back to this in a second, we use this one term pain as we do cancer, but we all know that cancer is a collection of different disorders that have different molecular substrates, et cetera. And the same thing's true of pain. Migraine pain is different than musculoskeletal pain, et cetera. And what are the different underpinnings that account for these different types of syndromes? What are their molecular mechanisms? Nociception, of course, is the process of detecting a noxious stimulus that leads to a pain response once that information is integrated by the central nervous system. These cells detect noxious stimuli at the periphery, for example, transmit this information to the spinal cord, the dorsal horn, and then through ascending spinothalamic tract neurons, transmits this information to the brain. Ergo, the old saying, no brain, no pain, because it's really a complex cognitive response. But we've been interested in, quote unquote, the simpler end of this, namely understanding how these stimuli are detected at the periphery. And these neurons that do this, the so-called nociceptors, or the primary afferent nociceptors, are really amazing cells because, as you'll hear this morning, they have to detect a wide range of stimuli, not only chemical stimuli, such as environmental irritants and endogenous inflammatory agents, but also physical forces like temperature and pressure. And they have to integrate all this information so that the gain of the primary afferent changes in the context of normal acute pain or injury-enhanced pain hypersensitivity. So we're really interested in how this process occurs. And as Ardem told us yesterday, actually, in many cases, these nociceptors act on their own. They're bare nerve fibers that detect stimuli to the skin or visceral or vascular target organs. But sometimes they also engage and interact with sensory-type cells within their environment and form sort of a partnership with them. And that's kind of a little bit of what I'll talk about today. So getting back to this issue about chronic pain, what is it? Well, chronic pain constitutes a collection of clinically and mechanistically distinct syndromes. So as I said, lower back pain is different than arthritic knee pain or post-herpetic neuralgia from shingles, et cetera. And one of the goals is to understand what makes these different pain syndromes distinct, because the idea is that if you can understand them in a distinct way, then you can target therapies pharmacologically or otherwise to more effectively treat different pain syndromes. So in my lab, the goal is to understand the molecules that are involved in pain sensation and temperature sensation, and to use structural and biophysical methods to understand how they work. And another goal is to choose one or two interesting scenarios or models that tell us about these specific pain syndromes so that we can go a little deeper into the physiology. And today I just want to tell you about some work that we started doing a few years ago that's pretty recent, that really relates to an aspect of somatosensation interoception, namely how we detect, and in this case pain, how we detect noxious stimuli within the body. And we're interested in an aspect of that, namely visceral pain. That is pain that might be associated with something like inflammatory bowel syndrome. And we've chosen this because it affects a very wide percentage of the population, up to 15% of people. IBS or gut pain can be post-inflammatory or post-infectious. In fact, the most common cause for instigating, the most common instigator of IBS, inflammatory bowel syndrome, and related pain is a bout of food poisoning. Another interesting part of this is that there's a big gender disparity. Women report suffering from IBS and IBS-related pain about 2 or 3 times more frequently than men. And you know this because if you see any commercials, at least in the States where they can advertise drugs directly, they always feature a woman young to mid-age. It's never a man. And so that says something about the target group. And we're also interested in this because some of the work I'll talk about is done with my wife. She's here today, Holly Ingram, who's interested in understanding sex differences in physiology. There's also a lack of biomarkers or overt pathology to understand visceral pain, and the sensory mechanisms remain obscure. But another reason we're interested in this is because it really highlights the interaction of the pain pathway with a bunch of different important factors. One is inflammation, as well as the microbiome, how this feeds into stimulation and hyperstimulation of the peripheral nervous system and sensitization that can actually be quite long-lasting. So after someone develops IBS-related pain, this can stay with them for days, weeks, or the rest of their life. And ultimately, how this leads to central sensitization, what happens to changes in the wiring at the level of the spinal cord or brain to instantiate this in terms of long-term pain pathways. So this work was done by a group we call the Gut Group, and was started by Nick Bologna when he was in my lab. He now has his own lab at Harvard, and Ryan Mori, who's now in biotech. And then more recent folks who've joined the lab, Koki Tuhara, Nathan Rosen, and Eric Figueroa in my lab, Archana in Holly's lab, and then Jim Bayer, who started off as a clinical pediatric GI fellow with Holly and now has his own group. And a very important contributor to this is Stuart Brierley, our long-term collaborator at the University in Flinders and Adelaide, who runs the visceral pain unit there. All right, so let me tell you a little bit about the cell groups that we've looked at in this study. So within the gut, say in the small intestine or in the colon, there's a lining of epithelial cells that line the villus of the gut that separate basically the cavity of the gut where all the food goes through from the nerve plexi, the vascular system, et cetera. And it's a thin, unicellular monolayer of cells that acts as a boundary. And within that unicellular layer, most of the cells are unspecialized, just epithelial cells that form a tight junction. But about 1 to 5% of the cells are specialized sensory cells, the so-called enteroendocrine cells. And among these enteroendocrine cells, there's one particular cell type that we've been fascinated by called the enterochromaffin cells. So EC cells represent about 1% of the total epithelium in this, that separate the lumen from the inside of the gut. And these cells actually manufacture about 90 to 95% of the serotonin in the body and are responsible for peripheral distribution of 5-HT. And they talk to a number of different cell types, but a prominent cell type with which they interact are these primary afferent nociceptors, in particular a subset that express one of our loves, the capsaicin receptor, TRPV1, but also express the one member of the serotonin receptor family that's a ligand-gated channel, an ionotropic receptor that my lab cloned some years ago called the 5-HT3 receptor. So this is, we think, part of the initial pathway that signals between the EC cell and the primary afferent nociceptor. These cells, these EC cells, are really beautiful, excitable cells, which Nick showed. Here you can see one bursting that's been loaded with GCamp reporter. As I said, these are polarized epithelial cells, but they're also excitable. They express voltage-gated sodium channels, potassium channels, and calcium channels. And Nick and Jim did a lot of work in a paper we published in Cell a few years ago, showing that these cells are sort of endowed with a number of detectors of noxious stimuli. These include, at the luminal surface, a TRPA1 channel, which we've worked on for years, which is we call the wasabi receptor, which has as its main goal to serve as a chemonociceptor to detect noxious chemicals. So when you eat wasabi and it goes through your gut, it's activating this receptor on these cells. And as well as other receptors, this olfactory-like receptor that is activated by a compound that I'll talk about called isovalerate, which is a short-chain bacterial fatty acid that's an irritant that's produced at high levels in people who have inflammatory bowel syndrome and other inflammatory diseases. There's also adrenergic receptor on the basolateral side that we are interested in because we think that it may participate in the communication between incoming sympathetic neurons that we know regulate gut activity, particularly under conditions of stress. So the questions we want to know are what stimuli actually activate these cells in vivo, what effect do they have on visceral pain and hypersensitivity, is this interaction between the EC cell and the primary afferent actually important for generating visceral pain, and what's the feedback from, for example, sympathetic input through the release, or sensory input through the release of neuropeptides, ATP, or norepinephrine from the sympathetic cell. So what's the dynamics of this interaction? And again, how does this contribute to visceral pain? So again, the questions are, does the EC cell activation is it sufficient to produce visceral hypersensitivity? What role does this play? And is it essential? Is it required for expression of visceral hypersensitivity, both in the short and the long term? Are there sex differences in this EC cell-mediated sensitization pathway? And can chronic EC cell activation produce visceral hypersensitivity in and of itself? And so these are some of the questions that we've looked at that I'll give you some. This is sort of a work in progress, so I'll give you a little bit of a work in progress in this regard. All right, so there's really, one of the nice things about studying this system, it's a little bit like studying cutaneous pain. There are a lot of great preparations that you can use where you can look at the activity of the nerve fiber and the EC cells, both in ex vivo explants, such as equivalent to a skin nerve preparation, in awake animal, in whole animals, et cetera. So this is one prep, sort of the most reductionist prep, where you take a piece of the gut and put it in a recording bath, and you can poke or prod the receptive field mechanically, you put chemicals on, and then you can record from the primary afferent nerve fibers in this chamber and look at spike activation and look for sensitization or acute activation that way. And here's an example of that. When you do that and you poke or prod this receptive field with a mechanical stimulus, you see spiking. And when you put on this compound called isovalerate, this short-chain bacterial fatty acid that we know is an important factor for mediating visceral hypersensitivity in vivo, you see that in fact you enhance the spiking. And moreover, this spiking that you see can be diminished if you put in a drug called elocitron, which is a blocker of the 5-HT3 receptor on the quote-unquote postsynaptic side of this interaction on the primary afferent nerve terminal. So this sort of helps to substantiate the fact that this pathway of the EC cell signaling through a serotonergic synapse to the primary afferent nociceptor is in fact a bona fide physiologic pathway that's relevant to activation of nociceptors within this system. Now, the other way to look at this system is to use intravital imaging, where you just use a mouse that expresses a GCAMP reporter, for example, in the primary afferent cells, and then you can stimulate the pathway by using intracolonic stimulation and look at the dorsal root ganglia and ask if those cells are activated. So here's what that looks like. Here you have a mouse. This is work actually done in Stu's lab, where you express GCAMP in the sensory in the body of the sensory ganglia. And in this case, he's instilled isovalerate into the colon of the animal, and we can look at activation of the nerve fibers in this more in vivo preparation. And one of the things that we found that's actually quite intriguing is that females seem to have a much higher baseline activity than males in this preparation in terms of their acute activity. And therefore a differential sensitivity to hyperstimulation by isovalerate. So here's sort of a raster plot of what that looks like, and you can see what males look like. In females, there's a lot more ongoing activity, and you can see that the number of neurons with basal activity is substantially increased in females compared to males, but the number of neurons that respond to the administration of isovalerate is diminished. And we think that's because females start off at a higher baseline, and so there's less of a dynamic range for sensitization. And so this is something that we'll come back to. And we're not exactly sure what's going on in this circuit, but that's at least a working model for trying to understand this. All right, the other way that you can look at this pathway is more of behavioral sensitization. And I would say here we really need to develop some new approaches to look at this. But sort of the standard in the field at the moment is to look at a basically crouching behavior. So we all know when we have gut pain, we tend to do this, and you activate through a motor reflex in the spinal cord the abdominal muscles within the abdominal wall as a reflex towards visceral pain. And you can measure that by putting electrodes on these on these muscles and measuring their motor activity. And you can also, score crouching behavior in the animal. But basically, when you're recording these stimuli, this is the crouching behavior that we're looking at. And what you do is you insert a balloon into the colon and inflate it to activate the nociceptors within that. It's sort of like when people get a colonoscopy, essentially. And you can increase the pressure gradedly and then look at the response. And here's an example of what that looks like. In a male mouse, you see that there's an increase in the response with pressure, shown here in the black line. When you instill isovalerate into the colon, you can see this very substantial increase in the sensitivity, which you see in these representative traces, where the amount of activity in the muscle fiber increases tremendously as you instill an irritant into the colon, presumably because now you're activating this pathway of the EC cells then signaling through a serotonergic synapse to the primary afferent. In females, you see less of an effect of isovalerate, just as you do in the intravital imaging that I showed you, presumably again because there's this floor or ceiling effect where you have truncated the dynamic range of the response pathway. So one thing we want to do now is to use genetics to understand what is the sufficiency and the necessity of these EC cells in this pathway in terms of mediating visceral pain, and are we really looking at the right locus to be able to understand mechanisms of acute pain and pain hypersensitivity, and ultimately try and piece out what the differences are between males and females in terms of mechanisms that could possibly contribute to sex disparities. So we've used sort of, the usual kind of chemogenetic strategies. In one case, we've expressed a tetanus toxin, specifically in the enterochromaffin cells. A tetanus toxin prevents fusion of the synaptic vesicles and diminishes release, presumably, of serotonin from the synapses. And these images just show that we've targeted these cells appropriately. When you look at, if you take an organoid from these cells, produce a gut organoid from these animals, rather, you can see that you have diminished release of serotonin in response to an activator of this wasabi receptor called TRPA1. And we see the same result if we look at serum levels from these animals. What's interesting about this is that you still see a significant basal serotonin tone. So we think that there's a basal release of serotonin and a stimulated release in relationship to administration of irritants that activate the EC cells, and that these may be two pathways. And we're developing probes to be able to look at that at a single cell level to really understand what the release dynamics are in these cells. The other thing that we've done, of course, is to put DREAD receptors into these cells so that we could activate them chemogenetically at will. And so if you use something like CNO or DCZ as an appropriate agonist, you can activate this pathway. And again, when you do that, you see a bump up in the amount of released serotonin from these cells, suggesting or indicating that the chemogenetic strategy has worked. Now, when you do this and look at these animals in this first prep that I talked about, this ex vivo prep, where you can stimulate the receptive field with isovalerate or mechanical stimuli, what you see is that the ability in both males or females to enhance the mechanical sensitivity with isovalerate, which is generally higher in males than females, you see that the expression of the tetanus toxin completely abrogates that effect. So you need to have release of serotonin from the EC cells in order to have this sensitization by this inflammatory agent. Now, if you look at mice in vivo and you look at their pain response using this sort of EMG-like recording from the abdominal muscles, what you see is something quite interesting, and that is that in both males and females, you diminish the sensitivity to distension of the colon, but the effect in females is much greater than it is in males. And again, we think this is because the females already start out at a higher level, so if you abrogate that process, they have a much larger dimension to fall. And so you can see that the response is actually, the effect of abrogating the signaling pathway is much more significant in females than it is in males. And you sort of see the opposite thing when you activate these cells with DREAD receptor, and that is that in males, you can potentiate the sensitivity to a colonic stimulus, whereas in females, you get much less of a sensitization. So we think altogether these assays are showing us that this is in the ex vivo prep, and the same thing seen in vivo. So one of the differences between males and females, we think, is where the gain is set in the system and how much and whether the females are close to the hypersensitivity to begin with, and how much they can be modulated in the dynamic range in terms of diminishing or activating activity of the enterochromaffin cell. Now, one of the exciting things I think about this that we found with this system, so one of the problems in studying the system and understanding mechanisms of sensitization is that the way it's normally done, if you want to, say, recapitulate or mimic an IBS-like long-term inflammatory syndrome in a mouse, is that you instill an irritant into the colon, such as TNBS, which creates a lot of tissue damage and leads to a very fulminant inflammatory response. But as in people, within several weeks, that inflammatory response will normalize, and the tissue morphology will regain its normal structure. So the colon will, of course, is a highly regenerative structure, and indices of tissue damage are diminished or eliminated or resolved, and animals are still left with visceral hypersensitivity for days or weeks. But it would also be nice to be able to instigate this kind of a hypersensitivity in the absence of inducing any kind of tissue damage, so we could have a more proximal mechanism that tells us about that, and something that's a little bit simpler. And so what we asked was, what happens if you activate EC cells persistently over time? Is that sufficient to generate peripheral hypersensitivity in the gut? And can that be used as a more controlled model to model gut hypersensitivity and essentially IBS, long-term consequences of IBS in terms of pain? So what Jim did was to implant a transmitter so we could measure the behavioral aspects, the muscle contractions, and then treat with this in mice that express a dread receptor, and treat with this activator of the dread receptor, DCZ, for about 3 weeks, and then wash the drug out, and after several days come back and ask whether this animal retains hypersensitivity to the gut, and using this in vivo assay. And what he finds, and actually we have even more impressive data here, is that you maintain this visceral hypersensitivity after stimulation of the dread receptor has been stopped, and you have a washout period. And what we're trying to do now is to see what's the shortest time of activation of the EC cells that we can affect to instantiate this response, and what's the longest washout time? How long does this last? And can we really use this as a model, as a more reductionist model, where we're just activating one cell and its connection to the primary afferent, and use that as a simplified, more defined model to understand what are the mechanisms underlying these long-term changes in pain, and what are the differences between males and females. And so I think that's a sort of a very exciting outcome of this that we're now trying to mine to really understand mechanism. And then finally, one of the associated behaviors with people who have gut dysregulation in almost any direction is that they experience anxiety phenotypes. And this is sort of interesting to people who are really, of course, interested in this now very popular area of gut-brain interactions. And so we simply ask, or Sean and Holly's lab ask, whether we see increased evidence for increased anxiety in these animals. And so far, we've just used a very simple standard test of this elevated plus maze. And what we find, in fact, is that you see increased anxiety either way, whether you activate the EC cell or you inhibit it. And here you can see some of those data, at least some of our early data, where you take a dread animal and you activate the pathway with DCZ, and you see that whether you measure distance traveled or time spent in the open arms, that these mice develop actually a rather substantial anxiety phenotype. And importantly, this anxiety phenotype can be reversed by giving animals this allosetron, which is the blocker of the postsynaptic 5-HT3 receptor on the primary afferent that's talking to the EC cell. And as I said, this turns out to be true also in these tetanus toxin expressing mice. Now remember, these mice are expressing tetanus toxin from birth. It's not an inducible situation. And you can see an increased anxiety in those animals as well. So I think this is telling us that no matter how you push the system from its normal set point, you see changes in behavior in these animals. And we'd like to understand more about that and understand the central pathways that are connecting the gut to the spinal cord and brain that are leading to this change in behavioral phenotype. All right, so in closing, let me just say that I think what we found is that EC cell mucosal afferent signaling is very important for driving visceral pain. This is a major locus for activation of the pain pathway, at least in response to some of the stimuli that we've looked at. There seems to be a higher basal EC cell activity in females compared to males, and this may contribute to sex differences that we see in the dynamic range of either acute or pain hypersensitivity. And I think excitingly, prolonged EC cell activation is by itself sufficient to recapitulate some aspects of persistent visceral hypersensitivity, and this now gives us a much more usable tool and easier and a much more reductionist tool to be able to understand the pathways involved here. And modulation of EC cell mucosal signaling elicits this behavior, cognitive behavior, commonly associated with functional GI disorders. And again, substantiating the idea that many people have worked on, that there's bidirectional signaling in the gut-brain axis. And the work that I've told you about has been supported by a couple of private foundations, but most of the work in my lab supported by, usually I say when I'm in the States, by you, the taxpayers, through the National Institute of Health, but anyway, by the U.S. taxpayers through the NIH. And I thank you for your time.
Wow, I mean, this was mind-blowing. Thank you, David. So we have actually quite some time for questions. Max, and then I see Soledad.
My question is whether the EC cells communicate with each other to propagate like a nociceptive input to neighboring regions of the mucosa.
Yeah, that's a very good question. You know, the distribution of EC cells is different in different places in the gut. You know, we've looked early on when we looked at EC cells and their properties, we've looked at the small intestine, but really what's relevant here, and we're focusing more on now, is to look in the colon, because that's, you know, the relevant part of the gut. They're spaced fairly widely apart, but, you know, the transmitters that they release, so one thing we're looking at is when they release 5-HT, what is the range of release? There's these old models that, you know, it's sort of like a sprinkler system and it's mostly action at a distance. But some of our histology suggests that the EC cell and the afferent actually can, in some cases, form more of a close synaptic interaction. So we want to know really what the spatial dynamics of that is. And so Koki in the lab is trying to develop methods to do that. But they probably release more than serotonin, including peptides. So there could be, you know, communication at a distance. But there's also other excitable enteroendocrine cells within the gut that more involved in things like satiety, et cetera. But this is an interesting question. How much crosstalk is there within the thin epithelium?
Also the feedback, the noradrenergic feedback.
Yes.
Bring the signal through the serotoninergic terminal
Yes.
to the central nervous system
And then back through the sympathetic system. And so we're very interested, for example, in what's the role of these adrenergic receptors on the basolateral side, and do they regulate excitability of the EC cell during the stress response? Yeah.
Hi, thank you very much for the talk. So you showed that the EC cell activation induces mechanical hypersensitivity, and I was curious about one of the plots that you showed where the spikes of stroke increased what seemed to me linearly with the mechanical stimulus. But when I think about pain, I think about a nonlinear process where something is amplified. So I was wondering if this assumption that I'm making is correct, and if yes, if this nonlinearity you think is happening at the EC level, cell level, or if it is down the pathway.
Right. That's a very good question. I think when you look at these, what we call VMR responses, the ones where you're measuring gut contraction, you know, you see mostly a linear response in terms of behavior. I think we're not sophisticated enough in these assays yet to actually be able to measure, to ask those kind of questions and measure the dynamics. That is a good question, and I think what we're looking at primarily, though, is recording from the nociceptor, and I think if you activate the nociceptor above a certain threshold, you will probably activate the pain pathway to some degree. But I think one other way to look at that is to begin to record from neurons within the spinal cord and see whether that linearity persists when you get to the second-order neurons, or whether there is a threshold that requires the activation of a minimal number of primary afferents coming into the dorsal horn, and that we haven't done yet. Yeah.
Thank you very much.
We have time for one more question. Keegan, please.
Hi, nice talk. Do we know if and how the chromatin changes in these EC cells after IVL exposure?
If the what? I'm sorry?
If and how chromatin changes after this acute IVL exposure?
Oh, chromatin, you mean in terms of gene expression?
Yeah, in terms of
yeah, we haven't looked at those at any sort of epigenetic factors or whatever. That's a good question. It's a little challenging to do. Well, you can, I mean, in many of these experiments to begin with, when we looked at the transcriptome, you can sort for the EC cells and pull them out. There's not many of them. We always tend to work on these cells where there aren't many of them. So it's like banging your head against the wall. But those, yeah, those kind of questions is I think that's one of the great reasons to try and develop this system, something like that, where we can activate the cells with a very defined agonist through a defined receptor and generate a hypersensitivity mechanism, because then we're not dealing with activation, for example, of complex immune responses in the gut by putting in an irritant like TNBS. So maybe with those kinds of systems, we'll be able to address those kind of questions. Yeah.
I'm sure there's tons of questions. I do have a lot of questions, but let's thank David for
Okay.
now.
Thank you.